All right, so let's go ahead and talk about gene expression today. This is a pretty modern idea in biology. So if you think about cells, cells are very busy little places, okay? And your cells carry the information to code for any individual protein that you need, okay? So like the cells in your toes can have the code to make, I don't know, nervous cells, okay? They have the code to make kidney tissue. They have the code to make whatever. How do we make it so that the cells in your toe, say like the skin on your toe, only grows that skin tissue, that epithelial skin tissue, and not kidney tissue, brain tissue, eye tissue? How do we make it so it only grows that particular type of tissue? Or cellular products, okay? Your adrenaline glands on your kidneys make certain hormones. Well, those are coded for by DNA that turns into a protein through transcription and translation. How come your toe doesn't make the hormones that your adrenaline glands make? Well, gene expression is the answer to that. We can't have every cell making every single protein on every single gene all at the same time. So this is kind of a complex issue because we want to turn on certain genes and turn off certain genes in response to certain factors in the environment. That might be, say you're a fetus, that might be grow, okay? And then it might be, okay, stop growing. That might be a broken bone. Grow new bone tissue only right here, okay? So we have to turn genes off and on in specific areas or else really bad things can happen, okay? There are certain genetic diseases where you grow tissue where you shouldn't, okay? There are certain genetic diseases where you don't make an enzyme or whatever, and that can cause the buildup of something in your body that's toxic. So it can be genes need to be turned on, or it can be genes need to be turned off. Okay, let's go ahead and talk about that. This is kind of summarizes what I just said. Both prokaryotes and eukaryotes have to be able to regulate which genes are expressed at any given time. If you're a tree, you don't want to grow leaves in the middle of winter. So what are the switches? There are things in the environment, and there are also things inside of the organism, okay? If you eat a bunch of candy like I just did, your body needs to make insulin to deal with that. So those genes will get activated. What else does this allow you to do? Okay, it allows in eukaryotes, multicellular organisms, it allows cells to become specialized. Okay, if you can only grow like one type of cell, there are animals like that. Sponges are very, very simple. They only have like one or two types of cells. You can put a sponge in a blender and it will reassemble itself, like because it only has one type of cell. That's pretty crazy. So you guys know what I mean by sponges, like SpongeBob SquarePants, like the kind that grow in the ocean. So it allows cells to be specialized. So if you think about like more complex organisms, like even something as simple as a worm has all sorts of specialized cells, okay? So it's not just things on the human level. Okay, let's talk about some things that are going to be happening in bacteria because they're very simple There's a lot of research done on bacteria because they grow very quickly and they're very easy to kind of like manipulate compared to like a big multicellular organism. So a lot of what we know about this starts with bacteria. Okay. We're going to start by talking about something called an operon. An operon is a gene group that can be turned off or on, okay? And operons are sort of an area where three things are happening. You have a promoter, okay? And the promoter is where RNA polymerase can attach. So remember, okay, we have to go back to the section we just talked about. RNA polymerase makes a strand of mRNA that's going to go out and allow your body to express the gene, okay? Maybe your body needs bone tissue. So that promoter will attach, that RNA will attach to that promoter and it'll start stripping off mRNA for you to make bone tissue, okay? We have another thing called an operator. The operator is sort of the off-on switch, and then you have the actual genes, okay? So that's pretty obvious. Now, operons can work in two ways. They can be repressible, okay? So the gene is normally on, and a repressor shuts it off. We don't need this right now, let's repress it, okay? So a normally on gets shut off, okay? The next type can be inducible. Inducible is, we don't make this unless we need it, okay? So it's always off, but if we need it, we'll go turn it on. There are certain things in your house that are always on. Maybe a certain light that you leave on. Maybe your, I don't know, what would be something in the house you always leave on? Your refrigerator is always on. So if you want to repress your refrigerator, you got to pull the plug and turn it off, okay? There are certain things in your house that you only use at very specific times, like say your garbage disposal. That would be inducible. You have to actually go turn it on, and then when you're done with it, you should shut it off, okay? So think about these as normally on, and then we're going to turn it off for repressible. Normally it's off, and we're going to turn it on for inducible, okay? These words are used in other areas of language. So if I say, we really need to repress that behavior, that means, hey, stop doing that, okay? If I say, we need to find a way to help induce you to do your homework, maybe I'll give you a candy bar every time you do it. So these words are found in other areas besides biology. Okay, everybody up to date? Good. So, there are regulatory genes, okay? A regulatory gene makes a repressor protein that binds to the operator to block RNA, okay? It's almost like you ever walk through the halls and you see that plastic shield in front of the fire alarm, right? You know what I'm talking about? And if there's a fire, you pull that thing off, okay? So the repressor protein is like the clear shield, okay? And it's going to stop RNA from grabbing on and transcribing the genes, okay? These are always expressed at low levels, okay? And it is reversible. We want to pull this off if we need it. Now, we have to rewind all the way back to, I think, Unit 3 and look at enzymes really briefly here. Okay, allosteric enzymes. This is just a review. So if you remember, This is a picture we used way back at the start of the course. Alliosteric activators. A substrate will bind to that site, and it stabilizes the enzyme so it remains open, active, and ready to go. So it's kind of like a switch for an enzyme. Once that comes in, the enzyme's ready to go, and the active sites open up. Otherwise, the enzyme's just hanging out, inactive, and dormant. So this is kind of a parallel situation. You also have allosteric inhibitors. So you have something that's always on in its active form, but when the inhibitor attaches to it, it shuts it down. So let's just look at those again real quick. You have allosteric activators, allosteric inhibitors, okay? Activators turn on, that kind of makes sense. It activates it. Inhibitors turn off. Everybody knows what inhibit means. All right. So these operons are repressible. There's a really well-known one that we're going to use as a perfect example. They call them illustrative examples in AP biology. And that's the Trp Operon. Okay? This operon in bacteria controls the synthesis of something called tryptophan. Okay? Since it's a repressible, transcription is active. So it can be turned off by a repressor. Okay? I want to repeat that. When a gene is repressible, it's on all the time until it's turned off by its repressor. Okay? It's always active until you shut it off, kind of like the refrigerator example. All right, I'm just kind of watching you guys to see if you're up to date. So here's how this works. Because it's repressible, we want to turn it off. Let's start with what it makes. It makes tryptophan. If you have a key level of tryptophan, remember set points? If a bacteria has tryptophan present, do you need to make it? Let's say it's reached its set point. Do you need to make more, or do you need to shut it down? You want to just keep making it for fun, or do you need to say, let's save some energy for something else? Okay, I see some heads nodding. If it's present, you want to repress it. Let's say all your tryptophan was gone. What would you want to do? You'd want to make more. Okay, so this is a gene expression example. We want to regulate the production of tryptophan. We don't want to make too much. We don't want to have not enough. So how does this work? Okay. The repressor is an allosteric enzyme that's only active when tryptophan binds to it. So when tryptophan is present, the allosteric enzyme is active. So here's how this happens. When there's too much tryptophan, tryptophan is more likely to bind to the repressor, making it active. Okay? So we're going to shut off the production of tryptophan when a lot of tryptophan is present by using this enzyme. So it's almost like a thermostat. We don't call for the air conditioner to turn on until it gets hot. We don't make the furnace turn on until it gets too cold. So this is a regulation method where we use the repressor to turn off transcription for tryptophan. And these can work in many different ways, okay? It can be it's always on and we're going to turn it off when we reach a certain level. It can be it's turned off and we're going to turn it on when we need it. Different signals are going to activate or inactivate genes. This is just a really simple example from bacteria. It can get much, much more complex than this. Okay. Take a look here. So here's the RNA polymerase at the top of this picture. Look on the top left, you see sort of that potato-looking thing that's on the promoter. RNA polymerase cannot slide down the gene because the repressor is blocking it. Tryptophan is present, so tryptophan turns on the repressor. Okay? Tryptophan keeps the repressor in place. All right? This is almost like if you've ever seen like I'm not going to go there. So tryptophan, as long as tryptophan is present, the repressor stays. Let's say the bacteria uses up all its tryptophan. The tryptophan will release from the repressor. The repressor is now free to go. Tryptophan kind of holds that repressor in place by changing the shape of the enzyme. When the bacteria gets low on tryptophan, the repressor moves out of the way and you start creating the mRNA for the bacteria to translate that mRNA into more tryptophan. This is really complex, guys. It can get really easy to get lost in this, but this is an advanced class. You're probably going to have to look at this more than once, okay? So just let's review it one more time. When tryptophan is present, it keeps the repressor locked in place. Let's say the bacteria goes into something in its metabolism that makes it use all that tryptophan. Well, that tryptophan is going to get used up. It will come off of the repressor. All right, now it returns to that state. Tryptophan is going to rebond with the repressor. The repressor is going to change its shape, and it will block the operator again. Now, does the promoter and operator actually get transcribed as part of the gene? No. Okay, we just start with the amino acids downstream of the operator. So you're looking at some more complex biology than you've probably ever looked at before. We cover gene expression in basic biology class here at our school at some level, but this is something that which genes get expressed at what time, this is your first time seeing it. They're already posted. All right, let's look at another type. Another one is the Lac Operon. Okay, the Lac Operon controls the synthesis of lactase. You've probably heard of that. Lactase is the enzyme that helps you break down the sugar lactose, okay? You've probably heard of people who are lactose intolerant. All right, so for this gene, it's inducible. It's turned off, okay? It's turned off. So how does that work? We have something called a Lac Repressor. It binds to the operator. So this one is going to turn on the opposite of the TRP, okay? So the inducer for the Lac Operon, for the Lac Repressor, is alliosteric, okay? So when it's present, it's going to bind to the repressor and turn the repressor off, and then the gene can be transcribed. Let's look at this one. Too quick? Just a second. Go back and think about the TRP one. When does it turn off? When tryptophan is present or when tryptophan is missing? It turns off when tryptophan is... No. Oh, yes. Okay, you're right. I meant the entire gene transcription. Yeah, you got to be specific. That's my fault. All right, let's take a look here. This one is activated when there is no lactose present, okay? This one is repressed when there is no lactose present, okay? So how does this work? An enzyme called alliolactose binds to the repressor and removes it only when that sugar is present. Because you don't need to make the enzyme to break down lactose unless lactose is present. Now, if you look at the two pictures, in the repressible one, it's repressed when it's present, okay? In the inducible one, it's repressed when it's missing, okay? I will make a little chart for you to kind of fill in to compare these two. I think the chart, like when is it there, when is it not there, is really useful. So this is something that you only need to make lactose in very special situations. So we're not going to turn it on until that situation is there. What is that situation? When you eat that sugar in milk. Okay, let's see if you can understand where we're going with this. Just a quick review of what we just talked about. Okay, when we compare these, the Lac Operon to the Trp Operon, how could you say they're similar and different, okay? Well, they both regulate transcription of genes, but one is repressible and one is inducible, okay? One is repressible, one is inducible. Now listen, these are like perfect examples. You may get test questions where they say they'll give you some really thick biological jargon, but you'll have to pick out, and it'll say, well, it's repressible when something is there or not there, and you'll have to interpret what's going on there. So a lot of people want to like memorize what's going on with TRP and Lac, and that's fine, okay? It's probably good to know those. But it's more important to know the difference between repressible and inducible. All right, what are the three parts of an operon? Okay, they are the promoter, that's where RNA polymerase attaches. The operator, that's sort of the off-on switch, and then downstream of that is the gene. So this is basically like, how do we turn off and on certain parts of DNA? All right, that was in bacteria. It's a little different in eukaryotes. So when we look at phenotypes, that's how things physically look, okay? So the phenotype might be, you know, for like, if we go back to Mendelian peas, the phenotype might be tall or short or something like that. So the phenotype of a eukaryote is determined by combinations of genes, okay? And not only the combination of the genes, but the levels they're expressed at, okay? So this is called differential gene expression, okay? Differential gene expression. Certain genes are going to be expressed at certain times and certain levels. And we don't want every gene to be simultaneously expressed. We talked about why. Just looking at the diagram, you can see this is a little bit complex. Okay, let's get into it. Eukaryotic gene expression has multiple stages. Things are a little bit more complex in eukaryotes. All right, in eukaryotes, chromatin and DNA are much more structured. In prokaryotes, it's just sort of a ring of looser DNA. It's not wound up as tightly, okay? So when we look at this, if DNA is very tightly wound up, it's going to be much harder for it to be transcribed. So this is a problem that prokaryotes don't really have, because their DNA is much more open and much more accessible. So, how do we take care of this problem? If you remember, DNA winds up around little particles called histones, okay? So, histone acetylation adds acetyl groups to histones that loosen up the DNA. If you want to reverse that, you use DNA methylation, okay? Methyl groups, and if you remember, those are just little functional groups from biochemistry. These cause DNA to either loosen or condense, okay? Histone loosens, methyl condenses. All right, I'm going to go ahead and go if everybody says okay. Not yet? Stick with me, guys. I see a lot of people kind of zoning out because don't get so lost that you zone out. If I need to slow down, ask questions. I notice in the age of COVID, no one asks questions anymore. Nobody wants to talk. Okay, so the first thing we have to do if we want to activate a gene is to get to the gene. To get to the gene, we have to loosen it. To loosen it, we have to use histone. Now we're going to start talking about something called epigenetics. This is why certain genes are expressed at certain times in certain amounts. Okay. Loosening and tightening DNA does not change the DNA, okay? It just opens it up and closes it, okay? It opens it up and closes it. So these modifications to the chromatin aren't going to change your genes, okay? But they can be heritable, okay? So modifications can be reversed. This is why there are certain times that these can be activated or inactivated based on things other than your genotype. Some things in the environment turn them on. There's a really famous study with mice or rats where they can make them better mothers, and it's inheritable. So we can change their behavior, and it's based on this epigenetic inheritance. We'll look at that experiment here in a little bit. Okay. Once we modify the chromatin and we allow that DNA to be accessible, specific transcription factors bind to these control elements. Okay, so this is getting really thick, really technical. So there are sections of DNA that we once thought were junk. Okay, we look at the gene, we look at the DNA, and we're like, there's no genes there. There's nothing there. So, that DNA doesn't even do anything. We're now starting to learn, but there are sections that don't really serve as a plan for a protein, but it's more of a binding site to say, start making this gene or stop making this gene. So gene expression can be either increased or decreased by opening or closing the DNA and binding to these activators or repressors, okay? So this is way more complex than bacteria, way more complex. All right, so let's look at how the RNA is processed. There is what's called alternative splicing of pre-mRNA. Okay, how does that work? Translation can be activated or repressed by things called initiation factors. And what are these? They are microRNAs and small interfering RNAs can bind to mRNA to break it down or block translation. You got to remember, when you make mRNA, it degrades. This is one of the reasons why the coronavirus or the corona vaccine had to be kept super, super cold, because without that cold, the mRNA breaks down. So if you remember, we heard some cases where, somebody either intentionally or accidentally didn't let a refrigerator work, and the vaccine is no good anymore. It's because the mRNA in it breaks down. Now, there are other types of vaccines that don't require that super cold. This is the reason why right here, because mRNA degrades. Okay, so a big difference between prokaryotes and eukaryotes is embryonic development. Okay, prokaryotes, every single cell is identical. In eukaryotes, your body has all different kinds of tissues. Okay, so cell division has to differentiate. during embryonic development, you develop 3 layers, and out of those 3 layers, all your basic tissue types arise. Embryology can be really complex stuff because you study when these different tissues arise, problems that can go wrong with them. This is called morphogenesis. Like, why does an organism have the shape it has? Like, why do you have 2 arms up next to your head, 2 legs at the bottom? Sometimes that doesn't go right. I just saw in a biology post, a puppy was born with 6 legs and 2 tails. Very, very rare. I see somebody shaking their head. Did you see that too? Okay. Anyway, when things like that happen, it's usually a problem with morphogenesis. Okay. So, how does this work? How do we differentiate between the different tissue types that a eukaryote's going to need? So, there are some things that are called cytoplasmic determinants, and these are substances in the egg cell that influences cells to differentiate into these different tissue types. There's another process known as induction, and that involves cell-to-cell signals that cause genes to change their expression. One of the first things an embryo does for a group of animals called Deuterosomes, and that's basically most things with a backbone. It makes this little opening on one side of the cell, and that eventually becomes the anus, okay? And so almost anything that has a backbone does this. There are some animals that eat through an opening, and then when they're done, and they just expel their waste out of the mouth opening, okay? Most animals are just a tube inside of a tube, okay? You have a tube that goes through you. It starts at your mouth, it ends at your butt, okay? Almost every animal is like that, okay? If you were in my anatomy class, when you open up our specimens, they're basically just a tube full of organs, right? They've got some attachments on them, but most organisms are just a tube in a tube, like a worm, okay? And so one of the things that we see are when do these openings form? And it's very, very early in embryonic development, and it's controlled by these mechanisms. So, how does this work? These 2 processes influence which cells are grown at what time to make a body plan for different organisms. These are called Hometic Genes, and they map out body structures. Scientists have researched these by making fruit flies, especially, like, grow extra parts of their body, grow extra limbs, grow extra wings, don't grow wings, don't grow this. There's some pretty scary looking fruit flies out there because they're eukaryotic. Okay, here's another place we see this. Apoptosis is not just part of cancer, okay? If you've ever seen pictures of embryos, you'll see the little baby sitting there, and its hands will kind of look like they're all merged together, almost like dolphin fins, okay? And so during fetal development, in order to allow these structures to sort of differentiate and to take form, we have to kill the cells between your fingers. And there are occasionally people born where that doesn't happen, and they're born with like what they call webbed hands. All their fingers are grown together. So certain genes turn on apoptosis during eukaryotic development. This also happens with a structure in your heart that bypasses your lungs before you're born, okay? Before you're born, you don't need lungs. So blood goes around your lungs. Fetal circulation is different from when you're born to before you're born, okay? Because before you're born, you're using an umbilical cord. As soon as you're born, that umbilical cord is cut off and you have to change your circulation. It doesn't happen instantaneously, but it does happen. There's a hole in your heart that goes from your left to right atrium. And that hole should close up after you're born. There are occasionally people where it doesn't, and they can have fitness problems because half their blood is deoxygenated. That opening is called the Foramen Ovale. All right, that was pretty thick stuff. We're going to go ahead and